So far, we have covered the process of transcription, and we've talked about how the cell uses the code in the DNA to produce a messenger RNA code. From here, we need to talk about the process of translation. And there's two ways that we can think of this. One is the actual mechanics, which will be the next video. And the other is how the translation actually happens, how we get from a message coded in nucleic acids in the messenger RNA into a physical protein coded by amino acids. And the answer is that we do this by a code molecule called a transfer RNA. And transfer RNA uses the sequence of the messenger RNA to bring in the appropriate amino acid. So the genetic code determines how the nucleotide sequence actually produces a protein. And there's a few different ways we can think about this process. Initially, it was thought of as a one gene, one enzyme hypothesis, or one gene, one protein hypothesis, meaning that any single DNA gene coded for one particular enzyme. And then researchers began studying this and found that it might be a little bit more subtle than that. There might be some more options besides just one enzyme. So we know that one of the major ways that we express a protein is by enzymes, by an enzyme that does a particular job in a cell, usually some sort of chemical reaction. So whether that's building another protein or a step in a metabolic process. So we can think of this as one gene, one enzyme. But really, we have more than just enzymes that are produced by a cell. There's lots of different proteins that can be produced. And so to be a little bit more specific, or in some sense a little more broad, we could say, well, we get one gene to produce one polypeptide. And that's really great, but what about those functional RNA molecules? In which case we would need one gene for one RNA. And in any of these cases, we get a phenotype. Proteins are polymers, which are molecules that are made up of many repeating units of the same type. So the repeating units in a protein are amino acids, and the amino acids are linked in a particular direction by a covalent bond called a peptide bond. Just like DNA had a direction and we could only add a nucleotide to one end of the molecule, proteins are the same way. We link our proteins in the same orientation each time and in the same direction. And the end result of translation is an amino acid chain, a linear chain of amino acids called the primary sequence. Now that's not the end of our protein journey. Once the protein is made in the cell, it's going to be folded and processed into its final form. And that final form can be one of several different types of biological functions. The most common function that we talk about for proteins are enzymes. These are enzymes that control the chemical activities in our cell, including our metabolic activities and all of our building activities, building DNA, building RNA. We also have proteins that are involved in gene expression. Those are specifically going to be the ones that help build those molecules, like our DNA polymerase and RNA polymerase. And we've got a few others on this list. You can find this list either in your study guide, handed out in class, or posted in Brightspace. I believe that our book has a small section on it. This is actually a summary from a different textbook, so you may not be able to find this exact list, but if you can't, let me know and I'll give you a copy. All of our amino acids have the same basic structure. In the center of their structure is a carbon molecule that we have called the alpha carbon. This alpha carbon is also called the central carbon because there are 4 different groups coming off of this carbon. Off of the top here, we have a hydrogen. So anytime carbon's not bound to something else, it's probably bound to a hydrogen. On the left-hand side, we have the amino group. So this is where we get the amino part of the name. Amino means that it is nitrogen containing. So we've got this nitrogen group and amine group that is nitrogens bound to carbons. Now, nitrogen is highly polar, so it likes to bring an extra hydrogen to tag along. So the amino group often has a positive charge. On the opposite side of the alpha carbon is the carboxyl group. Now, carboxyl is a COOH. It is a carbon double bonded to an oxygen and a hydroxyl group on the side of it. Now, a carboxyl group is an acid, which means it's going to release its hydrogen. And so it usually has a negative charge on it. And that's where we get the acid part of our name. So on one side, we have an amine group. On the other side, we have a carboxyl that becomes an acid. Now, the part that's different for all of our amino acids are the radical group, or the R group, that is a side chain off of our amino acid base, off of that alpha carbon. And that R group is going to differ a little bit depending on which amino acid is attached. Our amino acids are going to be joined together by a peptide bond. A peptide bond is a covalent bond. And so we are essentially going to make kind of a backbone, just like we did with our DNA. And that backbone is going to be nitrogen, followed by the alpha carbon, our carboxyl carbon, and then another nitrogen group. So this bond here, the C double bond to the O, bonded to the nitrogen, is the peptide. And that's going to form every time we add another amino acid. And so if we add an amino acid to this carbon here, we lose one of those oxygens. The other one is the double bonded oxygen that branches off, and then we attach our next amino acid in our chain. So the goal of the ribosome is to build the primary structure. The ribosome in translation is going to form that peptide bond between amino acids. And once that bond is formed, then our protein is going to take on higher level structures. Remember that when we link together our DNA, it could then be bound across the strands to a second piece of DNA, which forced that twisted ladder shape. And that's all based on the chemical angles of the bonds, and it's based on interactions between our nucleotides and the angles that they interact. Our higher levels of protein structure are similar. Each level of protein structure has particular interactions that they undergo, chemical interactions, either covalent bonds or hydrogen bonds, attractions between other molecules within the amino acid chain. So our secondary structure is either going to be what we call an alpha helix or a beta pleated sheet. So our pleated sheet looks kind of like an accordion. And then our next level structure is going to be those secondary structures folded back on themselves. And the way that they fold depends on the interaction of the R groups. And then for some proteins, we can get multiple tertiary structures associated together to form a larger protein. So for example, this is hemoglobin, and it is made up of two types of tertiary structures and two different types of tertiary structures to make a bigger quaternary structure. Now, the folding is going to begin as soon as that messenger RNA comes off of the ribosome. So as soon as it comes out of the ribosome, it's going to start folding and interacting. And then once all of that takes place, the cell will make some modifications. In Biology 101, we learned about the Golgi apparatus and the rough endoplasmic reticulum. So some refining of the structure is going to take place before it is loaded into a vesicle and released out of the cell or moved around to the cell to wherever it needs to go. So our genetic code is the code that we use to translate from our language of nucleic acids to our language of amino acids. Now, this genetic code is nearly universal. There are some minor exceptions where different codes code for different amino acids. But for the most part, it's universal, which is why we can express genes from, say, a pig in an E. coli, or a firefly luciferase gene in E. coli, or in a mouse, because this code is universal. The triplet code works for almost everything. So we call this genetic code a triplet code because every set of 3 nucleotides specifies 1 amino acid. In addition, there are several different codes that may code for the same amino acid. We'll talk about why that is in just a little bit. So there are 64 different possible codes or codons, and there are only 20 amino acids. So there are multiple codes for any single amino acid. With one exception, all of our amino acids are coded by at least 2 different codons. That one exception is methionine, which is a start codon. And there's only one code for methionine. Now we call it a start codon because at the beginning of every single protein that we make, we are going to find the code for methionine. And so methionine is going to be the first amino acid on every single protein. But it is a special amino acid called F-methionine. And we'll talk about that one a little later. If we encounter methionine code later, it just pops in a regular methionine. There are also 3 stop codons. So these are codons that terminate the amino acid sequence, and that leaves 61 codons for the rest of our amino acids. If we look at some of these, let's look at leucine up in the top left. Leucine is coded by 6 different amino acids. We have another one. Where did it go? I have lost it. Oh, serine. Serine, coded up here and over here, is also coded by 6. The remainder are either 2 or 4s. So valine down here on the bottom, for example, is coded by 4. Lysine coded by 2. So there's no particular set number or requirement. The other odd one is isoleucine. Not really odd, but it is only coded by 3 because one of those codes in its group is for methionine. You do not have to memorize this for homework or for a test. You will be given a codon chart. Although it might be helpful to memorize AUG because you're going to see it a lot. So there are several types of codons. Those sense codons code for amino acids. Initiation codon is always going to be AUG. It codes for the beginning or the start of the amino acid chain, but also codes for methionine if we encounter it in the middle after that initial methionine. And then we've got our 3 termination codes. The codons are written in a 5' to 3' direction, and they are read in a 5' to 3' direction. Because there are multiple codons for a single amino acid, we call our code degenerate. Now this just means that there are multiple ways to code for a single amino acid. So if you look at your codon chart, you will see that we have multiple codes for every single one of them. We have at least 2 besides methionine for all of our amino acids. We call these codons that code for the same amino acid synonymous codons. And this degenerate code actually leads to the ability of our tRNAs to take on a special function. So our tRNAs are transfer RNAs, and the job of the transfer RNA is to bring the appropriate amino acid to the messenger RNA at the ribosome. Because of the degeneracy in our code, our tRNAs are essentially degenerate too. What they can do is pick up amino acids based on just part of their code. So they can accept the same amino acid, but using a different anticodon. And so this allows the cell to more quickly have transfer RNAs bring in the appropriate amino acid. Those iso-accepting tRNAs exhibit something called wobble. So these tRNAs allow for the last base pair in the triplet codon to wobble or change. And even if it wobbles or changes, the tRNA can still recognize the appropriate amino acid and bring in the appropriate one. What this also means is that the tRNA can bring in an amino acid to the messenger RNA if the messenger RNA code is off at the last base pair. So if we look at this illustration here, we've got our transfer RNA. At one end, that transfer RNA has the amino acid. At the other, it has an anticodon or a complementary codon to an amino acid sequence. And if we look at the one on the left, this is the exact complement, AGG, UCC. The one on the right shows that wobble, and it's even got these little squigglies here on the side to show you that it can change. So as long as we have the AG part matching up, this tRNA can still bring in serine. So we could have the tRNA that is AGG insert the serine, or we could have the amino acid that it has an anticodon of AGA, and either one can insert a serine at that point. That AUG start codon is important for starting out our amino acid, but it has a secondary function, which is to establish the reading frame. So the reading frame is the specific set of triplets that are going to be read. And these triplets are read in sequential, non-overlapping, non-repeating patterns. So we are going to read 1, 2, 3, 1, 2, 3, 1, 2, 3 with no gaps, no spaces, no backpedaling. And so our start codon is always going to establish which frame we are on. And if you start at a different point offset from that AUG, then we're going to get a different amino acid sequence. So as an illustration, if we use a sentence instead, the red bat ate pie. Now our reading frame, the set of triplet letters that we read, is set by the beginning of the sentence, in this case, the capital letter T. And each of these words is a triplet. We're going to ignore the spaces in between, but each of these is a triplet. Now, if I start my sentence instead at the H, but still read my triplets, we get a completely different sentence. In this case, it means absolutely nothing. It's complete gibberish. If we start on the E instead, we get a different bit of gibberish. And so our proteins are the same way. We always want to start at the correct reading frame at the A in our start codon. Likewise, if we have an insertion into our DNA sequence, a base pair insertion or a base pair deletion, this is going to cause what we call a frame shift mutation, because it changes the reading frame. It might start off okay, but if we insert, let's say, something here, that B between red and bat, we get a whole new reading frame. We start out okay, but then if we continue to read in our triplets, we start to get nonsense.